The overall goal of this procedure is to evaluate with Piconewton sensitivity axonal adhesion to substrates. This is accomplished by first using lasers to capture and position a microsphere next to an isolated neuron that has a long neurite. The second step is to center the bead over the axon using an infrared laser. Next, the stiffness of the optical trap is calibrated by interferometric tracking the Brownian motion of the trapped bead before attaching it to the neurite. The final step is to take force measurement during delivery of UV laser pulses to lesion the axon. Ultimately, force spectroscopy is used to evaluate the release of tension produced by the axon lesion, which represents an estimate of the axonal adhesion to the substrate.
The main advantage of this technique respect to fluorescence-based technique is that it furnishes a physical parameter, the force, measured with Piconewton sensitivity, allowing a direct comparison of axonal adhesion to the substrate in different distinct experimental conditions such as chemical treatment or different kind of substrate for culture support.
To begin this procedure, place a glass-bottom culture plate containing neurons seated at 25,000 cells per milliliter onto the microscope stage and secure it into place. Then, focus on the sample and correct the position of the illuminating objective condenser to set color illumination, which will improve the bright field imaging quality and maximize the infrared or IR laser collection efficiency. Next, pipette a few microliters of coated microsphere at 10,000 microspheres per microliter into the culture dish.